Okay. So in this video, we'll be discussing more on enzymes, specifically enzyme inhibition. And before we can really discuss inhibition, we need to mention that most enzymes fall into two categories that will describe their behavior in terms of converting substrate to product. The first category that we'll discuss are the Michaelis-Menten enzymes. And the Michaelis-Menten enzymes are very similar to the enzymes that we mentioned in the previous video, in that the first step is to take the enzyme plus the substrate, form an enzyme substrate complex, and then convert the substrate to product. And so we follow this pattern where the enzyme binds the substrate to form the complex. The substrate is converted to the product and is released from the enzyme. So now the enzyme can come back and bind another substrate and go through the cycle again. And Michaelis-Menten enzymes are described using two numbers. And we won't mention how those numbers are found because it involves doing a lot of graphing and in quite a bit of detail. We'll just mention what those two numbers are: Km and Vmax. Km is the Michaelis constant. Vmax is the maximum velocity. Km essentially describes how well the substrate binds the enzyme, and Vmax describes how quickly the substrate S is converted to product P. And so essentially Km measures step 1 and Vmax measures step 2 as we go from substrate to product. And one way to help explain the difference between Km and Vmax and explain the difference between the substrate binding and the product formation step is, let's say we have a box full of oranges and you're told to take the oranges and remove all the peels from the oranges. Then the first step is to go reach into the box and pick up an orange. That's similar to the enzyme binding the substrate. You have to bind the orange with your hand in order to pick it up. And the second step is to take the orange in your other hand and remove the peel. And so that's essentially the second step. That would be described by Vmax. How quickly can you take the peel off the orange? And so that combination of how you pick up the orange and then peel it can be described by the Km and the Vmax. And it's similar to how an enzyme can pick up a substrate and convert it to product. And so these Michaelis-Menten enzymes can be inhibited or slowed down by inhibitors. And there are several different types of inhibitors. One way to classify inhibitors is reversible versus irreversible. And an irreversible inhibitor is an inhibitor that cannot be reversed. They bind covalently or permanently to the enzyme. And an example of an irreversible inhibitor is aspirin, or acetylsalicylic acid, that it transfers its acetyl group to the enzyme, and the enzyme becomes permanently modified and unable to work. And so it essentially turns that enzyme off forever. And the only way to restore the enzyme activity is to make new copies of the enzyme. Reversible inhibitors, they can come and go. And so they can bind to the enzyme and inhibit it. They can fall off the enzyme, so the enzyme can go back to functioning normally again. The inhibitor, it does not bind to the enzyme forever, but it can bind to it temporarily, fall off, and then bind back to the enzyme again. Reversible inhibitors are classified into two groups: competitive and non-competitive. And we'll get into the differences between competitive and non-competitive inhibitors next. So we'll begin with competitive inhibition. That we've already drawn this E plus S goes to ES goes to E plus P. And just to draw some pictures, we have our enzyme, and our substrate. I've changed the colors, but the idea is the same. That the enzyme binds the substrate to form the enzyme substrate complex. The substrate is converted into product, and that product is released, so the enzyme can come back and do the process over again. But in the case of competitive inhibition, we not only have substrate, but we have inhibitor, I. And so let's give the inhibitor a different shape. And the enzyme can bind the inhibitor. It has a similar enough shape that it binds to the same spot in the enzyme. So it forms this EI complex, this enzyme inhibitor complex, that competes for formation of the enzyme substrate complex. And so some of the enzyme will be taken away and binds to the inhibitor at any given time. And so there's less enzyme available to bind to the substrate and produce product. And so that's how we slow down the reaction rate. And so because the structure of the inhibitor tends to be very similar to the structure of the substrate when we have competitive inhibition, the inhibitor binds to the same active site. As substrate. And so that's why it competes for binding, because it occupies the same space. And because it affects the enzyme's ability to bind to the substrate, if we remember, the first step is measured by Km, the second step is measured by Vmax, then competitive inhibitors affect Km, but not Vmax. And so we can see that it only affects this first step, the binding step, by reducing the amount of enzyme available for binding. And so if we were to use our orange peeling metaphor again, where you're reaching in the box and you're taking the oranges and you're removing the peels from the oranges, that a competitive inhibitor would be if we took a bunch of plastic orange balls, the same color and the same size as the oranges, and we mixed them into the box with the oranges, so you couldn't tell the difference between an orange and an orange ball. And so every time you reach into the box, you might grab an orange, or you might grab an orange ball. And if you grab an orange ball, you have to put the orange ball back in the box and try again. And so that slows down your ability to take oranges and peel them. And so that would be like a competitive inhibitor. So now we'll discuss non-competitive inhibitors, which are different from competitive inhibitors. That we still have this pattern here, where the enzyme plus the substrate goes to the enzyme substrate complex, then goes to enzyme plus product. We still have enzyme binding to inhibitor, but we also have this enzyme substrate complex can bind to inhibitor, forming an enzyme substrate inhibitor complex, or our enzyme inhibitor complex can bind to the substrate, forming the same enzyme substrate inhibitor complex. And the way it works is, instead of having one binding site for both inhibitor and substrate, we'll have two binding sites. And so I'll redraw all of these enzymes with two sites, a triangular site and a circular site. And then I'll change our inhibitor from a square to a circle. So in the enzyme substrate complex, I'll put the substrate back. But we can also have the inhibitor bound in the enzyme inhibitor complex and the enzyme substrate inhibitor complex. And so with the non-competitive inhibition, I binds at a different site than S. And when the inhibitor is bound, we cannot convert the substrate to the product. And so we can bind the inhibitor at two different steps, either with the enzyme is alone or when the enzyme is bound to the substrate, that we can pull both the enzyme and the enzyme substrate complex down here to this inhibited state where we cannot produce product. And so because the binding is unaffected, but the reaction rate is still affected, it affects Vmax, but not Km. So it affects step 2, but not step 1. And to use our orange peeling metaphor again, that a non-competitive inhibitor would be like trying to peel oranges with a mitten on your hand. And so you can't grab the orange very well to get the peel off. You can still pick up an orange from the box, but you can't get the peel off the orange because you've got a mitten on your hand. And so this you would essentially have an enzyme substrate inhibitor complex. Because of the mitten bound to your hand, you can't get the peel off the orange, so you can't make the product. So that's it for the Michaelis-Menten enzymes. So we'll discuss the other class of enzyme. That would be the allosteric enzymes. And when it comes to allosteric enzymes, terms like Km, Vmax, competitive, non-competitive, don't apply, because it's a different type of enzyme. It follows different rules. That allosteric enzymes, they typically have multiple copies of the enzyme bound together. So rather than being a single protein, it has quaternary structure now. And so we have several subunits that are joined together to form an overall enzyme. And these different subunits can display a phenomenon known as cooperativity. And essentially what happens with cooperativity is that when one enzyme, one subunit binds a substrate, the next subunit is more likely to bind a substrate as well. And an example of this is hemoglobin. That again, hemoglobin has two alpha subunits and two beta subunits. Hemoglobin's not really an enzyme because it doesn't carry out a chemical reaction. It simply binds oxygen, but it does display cooperativity similar to allosteric enzymes. So when we get the first oxygen molecule bound to one of our subunits, then the other three subunits, their affinity for oxygen increases. And so now they're more likely to pick up another oxygen molecule. And once the second oxygen molecule is bound, then the cooperativity improves again. And a third oxygen molecule is even more likely to bind. And finally, after we have three oxygen molecules bound, then that fourth oxygen molecule has a really high affinity. And so the hemoglobin, that fourth subunit in the hemoglobin is extremely likely to capture a fourth oxygen molecule. And so as we go from 0 to 4 oxygen molecules, the ability of those subunits to capture that oxygen increases. And that's an example of cooperativity, that the subunits cooperate in how they bind the oxygen. But anyway, allosteric enzymes are oftentimes affected by allosteric effectors. And an allosteric effector can be positive or negative. positive allosteric effectors increase the reaction rate. And negative allosteric effectors decrease reaction rate. And similarly to what we saw with non-competitive inhibitors with the Michaelis-Menten enzymes, the allosteric effectors bind to other binding sites. They do not bind to the same binding site as the substrate. And so if we were to draw an allosteric enzyme, it might look like the enzyme I was drawing before, that we have the active site where the substrate binds, and we have the allosteric site where the allosteric effector binds. And so the positive effector, if it binds, it would make the enzyme more likely to make the enzyme work faster. If a negative allosteric effector binds, it would make the enzyme work more slowly. And several allosteric enzymes are subject to feedback inhibition. And in feedback inhibition, let's say that we have a certain biochemical pathway where a molecule A gets converted to molecule B, converted to C, converted to D, and finally converted to E. And each one of these steps is carried out by an enzyme, enzyme 1, enzyme 2, enzyme 3, enzyme 4. And let's say they're all allosteric enzymes, and finally product E comes back and acts as an inhibitor of enzyme 1. So it'll act as a negative allosteric effector of enzyme 1. And so the more E that we have, the slower A is converted to B. And so that reduces the amount of E that's produced ultimately. And so then as we have less E, then the inhibition is reduced and more A can be converted to B. But then we have more E, and so we have more inhibition, and we slow down the production of E. And so doing this, the body can control the production of this molecule E by using E to inhibit its own production by coming back and inhibiting one of the enzymes involved in producing molecule E. So that's an example of how allosteric enzymes work, and a little bit of how they're different from Michaelis-Menten enzymes. So we'll discuss a couple other things that can change enzyme activity. One is covalent modification. In that something can be covalently added to an enzyme or taken away from the enzyme in order to affect its activity. A common covalent modification is phosphorylation, in which a phosphate group is added to an enzyme, usually on a serine or a threonine. Sometimes other amino acids are phosphorylated as well. But when we phosphorylate an enzyme, we attach a phosphate group which has this structure to it, a phosphorus atom with 4 oxygen atoms, 2 negative charges. This wavy line is the protein that we're adding our phosphate to. And if we were to dephosphorylate the enzyme, then we would take this group and we'd remove it. And so essentially the phosphorylation and dephosphorylation can act as a switch. That if we need to turn an enzyme on, then we might phosphorylate it. And if we can turn that enzyme off by removing the phosphate group, then we can control the enzyme activity. And so if we have an enzyme, let's say when red, it's turned off, and then blue, it has a phosphate group attached, and it's on. So the phosphorylation step takes it from off to on by adding this phosphate. And phosphorylation is typically done by a separate protein called a kinase. And so there'd be an enzyme that has to come along and bind to this red enzyme and add the phosphate group to convert it from red to blue. And a second enzyme called a phosphatase can come along and bind to blue and cut off the phosphate to convert it back to red. So that would be dephosphorylation. And so using kinases and phosphatases, we can control the phosphorylation state of these enzymes and therefore control whether the enzymes are turned off or on or vice versa. Several cell signaling pathways involve phosphorylation, where like the protein A is phosphorylated, and once it's phosphorylated, it phosphorylates protein B, which can phosphorylate protein C, and so on. And eventually it triggers some other response in the cell. And so inhibitors of this kinase activity are important for controlling the cellular response. And there's a lot of research into anti-cancer drugs that help prevent the phosphorylation or dephosphorylation of specific proteins in order to try to kill cancer cells. Another class of another type of enzyme is called a zymogen, which is an inactive precursor to an enzyme. And so it can be modified to turn it into a functional enzyme. That many digestive and toxic enzymes are produced as zymogens. For example, the proteins trypsin and chymotrypsin, both of these proteins are responsible for cutting up other proteins. But in order to safely manufacture these proteins, your body produces them as a zymogen. And so if I were to draw that, instead of a typical Pac-Man sort of enzyme, it would have some additional part that would occupy or block the active site. And so this is an inactive enzyme because the active site is inaccessible. But we can cut off that segment, and now it is an active enzyme and capable of cutting up other proteins. And so it's a good digestive enzyme for cutting up proteins in your food so you can digest it. But you don't want it to be activated early because it will also digest your tissues. Another example is toxic proteins, like bee venom proteins. Bee venom phospholipase A2 is one that I worked with as a graduate student. And PLA2, it cuts up phospholipases, and it can actually drill holes in cellular membranes. And it can kill cells that way. That's part of what makes bee stings painful. It's also produced as a zymogen inside the bee, and when when the bee stings a person or whatever, the zymogen is another enzyme comes along and converts the zymogen into an active enzyme, and so the bee venom PLA2 goes from inactive to active, and now it can destroy cellular tissues inside the target rather than inside the bee. And so zymogens are a good way of controlling enzyme activity until you need those enzymes to be turned on. So that's it for our discussion of proteins and enzymes for this chapter.